So when sound is being transmitted via vibration from the stapes into the inner ear, we have the stapes that's going to be inducing vibrations on a anatomical region of the inner ear called the oval window. This is going to send vibrations into the perilymph. The perilymph is this dark yellow region, and then this endolymph is in this central region. So I want to make clear that the anatomy shown in the previous slide is shown here. So here, the oval window is touching the scala vestibuli. So the vibrations enter the perilymph of the scala vestibuli, and I want to show you that was over here, okay? And then they travel through there. So depending on the wavelength of the sound, the waves are going to travel through the perilymph differently. And they're going to, so for example, this is a 20,000 hertz sound, a high frequency sound. This is going to has vibrations hit the very beginning of the duct. Okay, so it's going to basically decompress this this membrane over here called the vestibular membrane, and then it's going to send the vibration into the endolymph, which is in the cochlear duct, and it's going to cause a deformation of the hair cells that are right over here. These little lines are the hair cells, and it's going to hit early. Now, if we look at a 2,000 hertz sound, the vibration is going to go further in distance down the vestibuli, right? And it's going to then put pressure on from the perilymph, will put pressure on the endolymph. It'll deform the hair cells, which will trigger action potentials. And then if we look at the 500 hertz, which is the lowest frequency sound, the shown in blue, it's going to hit further down. It's going to travel a bigger distance than the higher frequency pitches would do. So here we're getting the 500 hertz is going to go further, okay? And then it's going to be deforming the different hairs in different locations of the duct. So the point is that location is critical. We hear based on the location of the hairs that are being deformed and moving when these vibrations enter the ear. So again, the waves enter the perilymph up here. They're going to be pushing down on the vestibular membrane, which will then have vibration entering the endolymph. That'll be pushing down on those hair cells that are shown in the bottom here. And then they will then send action potentials to the central nervous system. And based on the location of those hair cells that are sending the action potential, the pitch of the sound will be detected and processed by the brain. So anyway, so this is the system where the vibrations are traveling through the scala vestibuli and also the scala tympani on the bottom. And then the vibrations need to go out somewhere, so they actually can exit out of the round window. So basically vibrations enter the oval window. They travel in the scala vestibuli. Then they will travel through the perilymph, inducing vibrations in the endolymph, which is in the cochlear duct. And then these vibrations can exit the scala tympani in the perilymph and exit in the round window. So they enter the open window, they leave in the round window. And I also want to point out that the vibrations will be entering on the vestibular membrane, but then where the hair cells are is on the basilar membrane. Okay, the basilar membrane is where all these hair cells are, and is the movement on the basilar membrane of the cochlear duct that we have all these hair cells. So the this region with the scala vestibuli, the cochlear duct, and the scala tympani, this little region here where we have the hair cells that can trigger action potentials when they move, that's called the organ of Corti. And basically, the organ of Corti, right, has inner hair cells, outer hair cells, and basically when this scala vestibuli is pushing down with the perilymph having vibrations, it'll then have the endolymph in the cochlear duct pushing down on the tectorial membrane and basically cause movement in the inner hair cell and outer hair cell. And it's these vibrations and movement that can cause potassium ion channels to open and rush in, and that can then trigger the action potentials. So I want to talk to you about two different kinds of hearing impairment. So one is conduction deafness, and this is where sound waves are not conducted from the outer ear to the inner ear. And this can be due to a buildup of earwax, too much fluid in the middle ear, damage to the eardrum, or even overgrowth of bone in the middle ear. And any of these problems impairs hearing all different sound frequencies. And hearing aids does help with conduction deafness. Another type of deafness is called sensorineural or perceptive deafness. And this is where nerve impulses are not conducted from the cochlea to the auditory cortex of the brain. And this could be due to damaged hair cells. So maybe someone heard a gunshot, right, without ear protection. And this can often only impair hearing of particular sound frequencies and not others. So, right, so if you have a really strong vibration that would hit, remember, the vibration is going to hit a certain location in the organ of Corti. It's not going to hit all the hair cells, it's going to hit hair cells in a specific location. So if the signal is too strong, that can cause damage. But it would do it at that specific location where the wave hits the hair cells, just like a wave coming in a hurricane could damage buildings that were on the coast in the area hit by the wave. It's really going to be a local effect. And again, so that's why it would be affecting some frequencies, but not all. And cochlear implants may be able to help. And presbyopia is the age-related hearing impairment where people's hearing is being lost as they age. So the vestibular apparatus is part of the utricle saccules and these semicircular canals that help us maintain balance and can detect movement. And these are part of the inner ear as well in the vestibular apparatus. So here the utricle and saccule are, and there are the semicircular canals. So these have sensory hair cells, and they also have the endolymph. And what happens is they can move, right? So in the cochlea, basically they move based on the vibrations of the sound. In the vestibular apparatus, we have a kinocilium, which is a really long hair cell, and then other stereocilia that are a little bit shorter. Okay, so at rest, if there's no movement, they're not going to be either depolarizing or hyperpolarizing. So if the stereocilia move towards the kinocilium, this is going to increase the frequency of action potentials, okay? That's going to be a major depolarization event, and you get very, very frequent action potentials. However, if the stereocilia are moving away from the kinocilium, there's going to be a decreased frequency of action potentials. And there is a basal frequency of action potentials that are occurring at rest. Okay, so you get a basal level at rest if there's significant movement in one direction. It's gonna, if they lean towards the kinocilium, we're going to see a much more frequent action potential. And if we have the stereocilia pointing away from the kinocilium, we're going to see the hyperpolarization and a much reduced frequency of action potential. So the other thing that we can have happen in the body is to detect when your head bends forward, for example. So the otolith organ is the utricle and saccule, and in there we have these stereocilia, which are these shown in purple, and there's these otoliths that create like a gel-like membrane that they sit in to kind of give it like a semi-solid state. So when this woman is leaning forward, what happens is that the gravity is going to be pulling on the stereocilia, and these otolithic objects and particles in the membrane are going to help it to sag, and basically the hair cells are going to be moving in one direction, right? And this is how part of how the body can sense. You can sense, if your eyes are closed, you can tell you're leaning forward and basically sense gravity. So the semicircular canals are along 3 planes to detect rotation. And as I showed you, they all have the endolymph on the inside, and the hair cells that are moving, the kinocilium and stereocilia, are surrounded by endolymph. And again, the stereocilia are in these gel-like tissues that give the membrane they're in some resistance to movement. So here's a picture of a man who's turning his head sideways, and I just wanted to show you how the hair cells can be detecting this movement, right? So here's the rest, right? You see the stereocilia going straight up, but then if he's moving his head, so he's moving his head in one direction, right? He's looking to the right, and now he's moving to the left. In the semicircular canal, there'll be movement of the endolymph, and what it's going to do is it's going to bend the cupola, right? It's going to start up here. It's more straight or vertical, and it's going to start bending. And it's the bending of the cupola and the hair, specifically the hair cells in the bottom, that are going to be detecting that, right? Because if the stereocilia are bending towards the kinocilium, we'll see more frequent action potentials, and if the stereocilia are bending away from the kinocilium, we're going to see a decreased frequency of action potentials. And then the central nervous system can process this information as movement, where are we moving? So the next topic is taste and smell. So this is an example of exteroceptors, right? And these are things that are, especially smell is something coming from outside of our body. And even taste is like taking an object from outside our body and putting it in our mouth. So taste responds to chemicals dissolved in food and drink. Smell responds to chemicals in the air. And we all know that basically smell affects taste.